Behavioral Neuroscience Chapter 6, Segment 4. Did sexual selection drive homid brain expansion? We can evaluate the rapid expansion of the human brain over the last 1.5 million years using Darwin's second evolutionary principle, sexual selection. Jeffrey F. Miller of 2000 suggested that natural selection to obtain food and shelter is not likely to account completely for the large brain and complex intelligence of Homo sapiens. The brain tripled in our ancestors between 2.5 million and 200,000 years ago, yet when studied they still made the same stone axe to survive. Only well after the human brain stopped expanding did technological progress develop. Brain growth did not correlate well with the supposed survival benefit of enlarged brains. He proposed an additional factor accounts for large human brains. Creativity, along with related brain growth, is due to sexual selection for abilities to attract attention, stimulate, and surprise potential mates. He hypothesized that sexual selection for artistry and creativity may lead to increased brain size and is supported by findings regarding the bowerbird family. To attract and impress females, the bowerbirds construct elaborate houses made of twigs decorated with colorful objects. They have large brains compared to the other birds. Box 6.3 is evolutionary psychology. Evolutionary psych is a field that views the human mind as a large collection of cognitive modules, each shaped through natural selection, to solve a specific adaptive problem. Ethical considerations prevent direct experimentation on many of the variables of interest, so studies tend to use indirect surveys and correlation designs. Researchers are now rethinking the notion that cognitive modules are exclusively highly specialized and are broadening the evolutionary view of psych to include the evolution of general-purpose mechanisms such as memory processes. Private species differ in gene expressions. Humans and chimpanzees can differ on a genetic basis in two principal ways. First, the DNA sequence of specific genes may vary in important ways between the species. Humans and their non-human relatives may also differ in how those genes are expressed and constructs a complex brain. An example, the ASPM gene influences the size of the cerebral cortex. Humans inheriting one version of the ASPM develop very small brains and are mentally disabled. The protein encoded by ASPM differs considerably between humans and chimpanzees, suggesting that the sequence of this gene evolved rapidly in the ancestor line leading to humans. When gene expression is compared in blood and liver cells, humans and chimpanzees are more similar to each other than either of them are to monkeys. When gene expression is compared in the brain, humans are more different than chimpanzees are from monkeys. The pattern of gene expression in the brain has changed and accelerated in the human lineage, presumably under selective pressure. Even a small change in gene expression can cause a dramatic difference in the brain development, and it may be what makes humans unique. Evolution continues today. In some cases, selection is driven by natural causes, and some by human behavior. An economically important case of recent evolution concerns commercial fishermen. In many regions, fishermen are allowed to keep one fish if it is over a certain size. Atlantic cod off the coast of Newfoundland have been maturing at smaller sizes over several decades, probably because the largest fish are the ones that are being captured. The remaining small fish can produce fewer eggs than the large fish, and the reproduced egg production can help explain the collapse in the cod production. Are we still evolving? The human brain is the product of selection forces encountered over the extended period of time in an ancestral environment thousands of years ago. Medical science has largely replaced natural selection in determining who dies and who gets to reproduce. Cultural evolution is the ability of humans to pass hard-won knowledge along to the next generation. In order to determine whether a candidate gene has been under strong natural selection pressure in the recent past, scientists look at the gene bundled together with its neighboring stretches of DNA, known as the individual's halotype. Minor variations where one nucleotide subsides from another can occur at certain locations within the gene, called single nucleotide polymorphisms, or SNPs. Most SNPs have two versions, which are called alleles. they cause the variation within traits. If one allele confers a slight reproduction advantage on those who possess it, it will come under natural selection pressure. Genes selected for will gradually spread throughout subsequent generations until most people possess the same allele. Sequencing a gene in a sample of people does not reveal whether the gene has been subject to recent evolutionary pressure. Not all changes in recent humans are evolutionary. Some are medical and some are environmental.